For cell and gene therapy, biologics, and nucleic acid-based medicines, the biopharmaceutical industry is entering a pivotal stage. What began as early-stage clinical promise is quickly scaling up to commercial-stage manufacturing. This evolution brings new demands, especially around quality control QC processes for characterizing and quantifying critical product impurities. We are issuing this executive report to highlight one such set of impurities, empty capsids and empty lipid nanoparticles (LNPs). We aim to provide an overview of the market opportunity, technology trends, and leading companies developing key detection and QC tools. Today, we examine the key players in the space, including Sartorius AG, Viralgen Biotech, AskBio, and NanoImaging Services. But first, let's review some overarching trends shaping this landscape. Empty capsids and empty LNPs represent significant, but often overlooked impurities in advanced biologic drug products. Their presence can compromise therapeutic efficacy, increase toxicity risks, and introduce major regulatory uncertainties. In particular, gene therapies using viral vectors like AAV commonly contain a mixture of full and empty capsids, where only the latter provide no therapeutic benefit while still eliciting immunogenicity. Similarly, mRNA therapeutics and vaccines formulated in LNPs often feature a heterogeneous population of full and empty nanoparticles, impacting both product potency and side effect profiles. To address this, industry leaders, contract manufacturers, and regulators are seeking to establish standardized methods for accurately quantifying these impurities. Though there remains a critical need for robust, sensitive, and scalable detection technologies. In fact, the biopharmaceutical industry is rapidly maturing from early-stage clinical trials to commercial-stage production. There is increasing demand for sensitive, robust, and regulatory-compliant detection technologies for quantifying empty capsids and empty LNPs in gene therapies, mRNA therapeutics, and other biologics. Several important market dynamics are converging to make this an attractive investment opportunity. First, there is clear market pull from the hundreds of ongoing clinical trials and growing commercial pipelines for cell and gene therapy, mRNA therapeutics, and other advanced biologics. Second, there is technology push driven by emerging analytical techniques that disrupt legacy detection methods like qPCR and traditional electron microscopy through greater sensitivity, speed, and scalability. Third, there is a need to meet stricter regulatory requirements and demonstrate batch-to-batch consistency, driving biopharma developers to adopt advanced analytics. And fourth, investors and strategic partners increasingly view robust quality control QC capabilities as critical for clinical success, licensing, and scalability. These factors are creating a unique market opportunity for innovative solutions that address the urgent need for efficient, accurate, and scalable detection technologies for empty capsids and empty LNPs. To learn more about our perspective on this market, access our full report on our website. Now let's review some of the leading companies poised to capitalize on this opportunity. Sartorius AG is a global life science and bioprocessing technology leader providing end-to-end manufacturing and analytical platforms. For cell and gene therapy, biologics, and mRNA therapeutics, they have developed proprietary QC tools for single-particle characterization, including optofluidic force induction (OF2I) technology and mass spectrometry-based systems for measuring AAV full-to-empty capsid ratios and LNP payload content. Sartorius is strategically positioned to capture high-margin business from biopharma and CDMO seeking to implement robust, scalable, and real-time QC solutions for their advanced biologic drug products. Its investments in high-resolution analytics and AI integration are reshaping how particle heterogeneity is assessed, moving from traditional time-consuming methods to automated high-throughput platforms. Moreover, its focus on developing and acquiring analytics companies positions it well for long-term growth in the CGT analytics market. Viralgen Biotech, a subsidiary of Bayer's gene therapy arm AskBio, is a leading contract development and manufacturing organization CDMO specializing in AAV gene therapy production. The company employs proprietary analytical ultracentrifugation AUC, charge detection mass spectrometry CDMS, and emerging real-time analytical systems to distinguish full from empty capsids. Viralgen and 𝗘𝗧𝗣𝗧𝗬 benefit from proprietary QC methods and technologies developed internally through years of gene therapy research and development. Their participation in standards-setting consortia and leadership roles in defining best practices and guidelines for AAV QC further strengthens their reputation as a quality leader within the gene therapy field. Furthermore, the companies are expected to license their proprietary QC platforms as the AAV market matures, opening up new revenue streams beyond their core manufacturing services. NanoImaging Services is a leading provider of high-resolution structural imaging and particle characterization services using cryo-electron microscopy cryo-EM and transmission electron microscopy TEM. The company supports GMP analytics for AAV and LNP-based therapeutics, offering visual confirmation of particle structure and payload distribution. NanoImaging differentiates itself through deep expertise in cryo-EM, coupled with AI-based particle classification and quantification, specialized software for complex nanoparticle analysis, and consultative partnering to help biopharma clients meet evolving QC needs. As demand for high-resolution analytics in clinical-grade manufacturing increases, the company is ideally positioned to serve as a critical partner to biopharma developers and CDMOs lacking internal imaging capabilities. In summary, these four companies are at the forefront of developing and applying innovative technologies for the detection and characterization of empty capsids and lipid nanoparticles in cell and gene therapy, biologics, and mRNA therapeutics. As the industry evolves and matures, the strategic importance of these QC capabilities will only intensify, presenting both challenges and significant opportunities for investors and biopharma developers alike.